Hello, students. Today we're going to look at the Punnett Square. So the Punnett Square is just one way of looking at probabilities that Mendel came up with for predicting what the gene pool would look like in the next generation. And as we discussed in class, we saw that he did a lot of scientific experiments. He actually counted his results and he drew conclusions and he looked at his results and analyzed them. So what we're doing today is we're going to look at how to make a Punnett square, and we're going to just do some simple stuff that students get wrong most of the time. So what we are going to start out with is, let's see if I can actually get my pen back. We're going to look at a monohybrid cross, all right? And a monohybrid cross means it is for just one pair of genes, one pair of genes. Okay, so one pair has two alleles, right? Because one chromosome will have one allele, and the other chromosome will have the corresponding allele at the same locus, right? So this is the locus, and that would be the allele, and this is a chromosome homologous pair of chromosomes. All right, so let's just say we're looking at just one locus on a chromosome. And let's say this one locus happens to be flower color. Okay, so we're going to start very simple, and we're going to see how this is very easily done with a Punnett square, because we don't want to write it out in a funny way. We're going to write it out in a really nice way. So let's say that the flower color is purple, like Mendel saw. And what he saw was that homozygous parents for purple produced homozygous baby plants. And what he also saw in the population was another color which was occurring very infrequently, and that was white. So he assumed that this color, or the gene, this would be the gene, is color, but there are two variants of the color, and this is on separate alleles. So on one allele, there's that, the other allele may have that. And as we looked in class, actually the white allele occurs because one enzyme is not produced, so the color is not produced. So what he did was he took homozygous parents of purple, homozygous parents of white, and he crossed them. And so what did he get? He got this new ratio of babies, or baby plants, and all of them were purple. Let me get my pen back. So this was the F1 generation. The parent generation was purple and white, and it gave to purple children. So he reasoned that, well, maybe the white has just disappeared. It's been eliminated from the gene pool. But when he crossed these purple babies with themselves, what he ended up with in the F2 generation was some purple, mostly purple, but he also got white. So the F2 generation reflected the grandparents. And what he saw was that this ratio is always 3:1. And what we can do now is figure out how did he do this, and how does this actually occur. And if it occurs for flower color, that's great. But if it occurs for other genes, because we're not just made of one gene, then how does that work? So let's say that the purple color is going to be represented by capital P, and the white, because it's the same allele. So please use the same letter of the alphabet, but lowercase. Okay, it can only be lowercase. And the dominant allele, so the dominant color, and this is the recessive color, because this is not the recessive color does not show in the F1 generation, so it's always going to be lowercase. So when we have parents, this is the P generation, we're going to cross them, and what we end up with is, let's say this is the dad, and this is the mom, tiny little one. Okay, so this is the female side, and that is the male side. And so what do we see? What kind of children are we coming up with? Remember, in meiosis, you just get one homologue of the chromosome. You don't get both pairs, but in the zygote, you get both pairs back. So here, the dad is donating a capital P, and the mom is donating a little p. And in this box, you get another capital P from the dad, and then you get a little p from the mom. And in this box, you get the same capital P from the dad and small p from the mom. And so this is the F1 generation, which is all completely heterozygous and all purple, because under the law of dominance, only the dominant gene will express itself in the F1 generation. All right? So notice that these are all hybrids now. They're no longer pure. They're no longer homozygous. They're now heterozygous children. All right? So what we have now, and I'm going to get rid of this, and we're going to go on to producing the next generation, no longer F1, but we're going to produce F2 generation, which is crossing these hybrids with each other. Okay, so here we are. We have another Punnett Square that we're going to be making, and we start out with like this nice big Punnett Square, so we can look at all the possibilities. Now remember, the parents are all hybrids, so it doesn't matter if you put the dad here and the mom here, but it will still produce these kind of gametes. And so let's cross the first zygote, and what you get is two capital P's, one from the mom and one from the dad. Then here's another possibility. So you get one capital P from the mom and a little p from the dad. All right? And then in this box, you get a capital P from the dad and a little p from the mom. And in this box, you get both small p's from both parents. And so when you look at the phenotype, what you have is purple. This flower will look purple because it's homozygous like the grandfather. It's completely dominant. Here you get another purple, and that's heterozygous like the parents of the F1 generation. And the third box is also purple, but this box is not purple anymore. So now this box is just going to be white, and this is going to be like the grandparents that we had previously. So what you have is a ratio of 3:1, 3 purples to 1 white. All right, and that is a monohybrid cross. The most important thing to remember is to use the same alphabet letter, all right? Do not use other letters like W for white and P for purple and then get it all confused, because it will not work. So it has to be the same letter. You just change the case. That's the convention, so people do not get confused. So going on to Mendel's second law, or going on to find out what happens if we have not one gene, because obviously chromosomes carry a lot of genes. They don't carry just one gene. And so what we have then is, let's pick two traits. And we are going to pick the traits that Mendel picked. So he said there were yellow seeds, and then he saw that they were round. Um, well, what he found was the yellow and the round, these were dominant traits. And then some seeds he saw were not yellow. Actually, they were green in color. And then they were not round in appearance at all. They were wrinkled. So these were recessive, and he did this by doing two Punnett squares to find out what was recessive and what was dominant by using just one trait at a time. So this was a monohybrid cross. So he figured it out. And then he decided, well, I'm going to look at two traits at the same time and see what happens. What kind of ratios do I get? So first we have to name these traits. And we're going to call it capital Y for yellow and capital R for round. So therefore, the green has to therefore be little yellow, and wrinkled has to be little r. Okay? So here we are. We're going to make another Punnett square, but this time this Punnett square is going to be kind of big and gigantic. So I'm going to, so let's see if I can try and figure this out and put this in here. No, I can't. So let's draw these parents first. So the first generation, they're homozygous. So that's one parent. And the other parent is green and wrinkled, homozygous. One parent is homozygous for yellow and round. All right, so this is the parent generation. And this is a dihybrid cross. And we're going to explore that. All right, so we're going to make a Punnett square this time, but it's not quite going to be the same. It's the same idea, but I'm just going to not do that. I'm just going to make a great big old Punnett square. And here, we're going to draw a nice big Punnett square like this. And we'll have actually not 4, but 16 cells in it, because our parents are like this. All right, so when meiosis occurs, what we see is that due to the law of independent assortment, this is going to be assorted independently from that, right? Yellow color and the seed appearance is going to assort independently. So we end up with like this, or we could end up with like this. We could, as you can see, there are all kinds of possibilities. So let me just write them all down. So we could have capital Y and capital R, capital Y and small y and capital R, small y and small r. And from the other parent, we get the same component. So when we cross these in the zygote, what we get is two capital Y's, two capital R's in the first possibility. The second possibility has two capital Y's, one capital R, one small r. And then this next possibility is this combination. And here we are with this combination. So we go down to the next row, and we find this combination, capital Y, small r, capital Y, big R, small r, capital Y, small y, and two small r's. And in this one, we have capital Y, small y, two big R's, capital Y, small y, one big R, one small r, two small y's. We haven't seen this combination before. And then two small y's, capital R, small r, and then big R, small y, capital R, small r, big Y, small y, two small r's, two small y's, one big R, one small r, and the last one is all small. All right, so let's look at these. What do these represent? Um, this first, let's go cell by cell and see what are these actually coding for. Because now we have it in code and we've got to say that this is, um, here we get, how is this going to work? Because we remember that, yellow is like this, green is this, round is this, and wrinkled is this. All right. So that anything that's under the lowercase is going to be can only express itself if it's in double dose or recessive. Two of the small letters together will make it express. Otherwise, it won't express at all. So let's see in the first cell. First cell has two capital Y's. So of course this is yellow. Oops. And two capital R's. So this is round. Okay. Next one is, why don't I just try and use another color? Yeah. So we're going to try and use yellow. And then we're going to try and use green. And this might take a while, but we could do this. We could actually do this. So let's say round is blue, and try using, well, not that. That's not gonna really look good, but we can use maybe purple. Does that show up? Yeah, it does. Okay, so what we have here when we try to look at this big Punnett Square, I know it looks a little scary, but it's really not. So we're going to go around, and instead of me writing all this stuff, we're actually going to go and put in colors. Okay, so I'm going to erase that, and we're going to put in the colors. We're going to go cell by cell and look at all the capital Y's. So here we are. This one is yellow. Yeah, yellow color. This one is yellow too. This one yellow? Yes, it is, because it's heterozygous. So is this. 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 Yeah. So is this. So is this. Not that, not that, but yes, this one. And this one. So what do we have now? We have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 12 yellow colors. No problem. Let's go and check and see what the other dominant trait is, and that was round. And we made that blue, didn't we? All right, let's go check all the blues. So round is going to be blue. This is going to be round too, yes, because it's heterozygous. This is homozygous round, so obviously that's round. This is heterozygous, another heterozygous, not this one, but this is round. Not that one, but this is round. And this one is round. This one is round, although it's not yellow. And this one is round. This one is round. And the last round we see is this one. Kind of cool, yeah? So now we're going to go and look at all the greens. Okay, so this one is homozygous recessive. Sorry. Oh, whoopsie. I'm messing up here. Let me just erase this and go to green, which is my pen over here. Let me pick the pen. Okay, so we're looking for a little yellow. Anytime I see a little yellow, I'm going to draw a green circle. That's one. Here's another. Here's another. Here's another. So actually, there are only 4 places I can dump the green color. Let's go and look at the purple color, which happens to be the recessive form of the seed shape, and that's wrinkled. And that happens to be this one, yeah, that one, that one, and that one. Okay, we're done. So now all we have to do is count it up. Okay, um, so let's see, how many yellows and rounds do we have? We have 1, 2, 3, 4, 5, 6, 7, 8, and 9. Okay, so we have 9 yellows. Let's see how many um, greens do we have. And we actually, so this is yellow and round. Now let's see how many um, yellow and, I'm sorry, green and round we have. So here's one green, okay? So that's one and round. No, it's not. Yes, it is. And here's another green that's round, and here's another green that's round. So we have 3 green rounds. And let's see how many yellows and wrinkled. Oh, let's get rid of these as we have gotten rid of, so we don't have to look at them again. And let's see how many yellows and wrinkles we have. So this is one, okay, here's another one, and here's another one. So that is also 3. So let's get rid of these two. And then we have the very last cell, and that happens to be just one recessive, double recessive cell, and that is given over here. So we end up with a ratio of 9:3:3:1. And that is how we come up with a dihybrid cross. Whoopsie. Let's see, what happened to everything? It kind of disappeared. Hmm. So that is how we come up with a dihybrid cross, and we find the ratio of 9:3:3:1 when we cross the generation that is a hybrid. So what we have here is Mendel's law showing you that the independent assortment of traits are occurring because when gametes rearrange themselves and reform chromosomes, it has nothing to do with one trait and the other. The traits all independently assort. And so you end up with a ratio of 9:3:3:1. And I hope that's helpful.